Hello, 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 hello. Am I audible? Hello, 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 hello. Am I audible? Hello. Okay. So, a very good evening to all. Welcome to Goscore Learning. We are the fastest edtech in GCC. Okay. So, welcome back again. Myself, Amala Anjos, and we will be continuing with the first chapter, that is the second part of this chapter. So, in the previous class, we have discussed about different topics on the first chapter, that is sexual reproduction in flowering plants. Yes, yes or no? Yes. So, in those portion, we have discussed about microsporogenesis, megasporogenesis, and also we have done with pollination, different agents of pollination. Okay, these were the topics that we have covered in the previous class. So today we will be continuing with the same chapter, that is we'll be continuing with outbreeding devices, then pollen pistil interaction, then we will be discussing with double fertilization. Moving into the next part, we'll be discussing with the structure of embryo, then seed, then fruit, then we'll be discussing with apomixis and polyembryony. These are the few topics that is remaining in this chapter. So it is very interesting to study about plants, right? Plants, we can see everywhere plants, and the study about the plants is also something what we can, it is interesting, right? Okay, so today we will be continuing with the first topic, that is outbreeding devices. Okay, so what do you mean by outbreeding devices? Okay, it is some kind of device. Is it some kind of devices? No, right? Okay, fine. So if you're considering a flower, mostly the flowers are hermaphrodites. Okay, what is the meaning of hermaphrodite? Okay, what is the meaning of hermaphrodite? We can say this hermaphrodite flowers are also known as bisexual flowers. Okay, they are also known as bisexual flowers. So what happens in bisexual flowers? Bisexual flowers means both male flower is present and also the female reproductive structures are present in the same flower. Okay, what is the condition here? Both male and female structures are present in a single flower. Okay, both are present in a single flower. So, dear students, there is possibility to self-pollinate. Okay, what is the possibility? The possibility is self-pollination. Last class we have discussed about what is self-pollination, right? It is the transfer of pollen grain from a single flower to the same flower to the stigma, or we can say to this pistil part. Okay, so if both male and female structure, if both male and female structures are present in a single flower, the chances are more for self-pollination, not for cross pollination. Okay, so in that particular plant, if you're considering a plant, you can see a small leaf, then flower, and we can consider this as a plant. Okay, what is the condition of this plant? This plant is bisexual plant. Okay, this plant is this flowers produced are bisexual flowers. Okay, all the flowers which is produced inside, all the flowers which is produced inside this plant will be bisexual flowers. Okay, so only the chance is self-pollination. And dear students, the self-pollination, continuous self-pollination, continuous continuous self-pollination will leads to a condition now called inbreeding depression. Okay, what is the condition? It leads to the condition called inbreeding, inbreeding depression. Okay, so if continuous self-pollination happens, if continuous self-pollination happens, what it leads to? It leads to inbreeding depression. Okay, that means the characteristic features of the genetic or the genetic combinations will be having some kind of mutation which is not favorable to that particular plant. Okay, so if you're considering this with the human beings, see, consanguineous marriages, you know that, right? Cousin marriages. If cousin marriages are being done in a same family, the chance of getting genetic disorders are high. That you have learned, right? So the same happens in the plants too. If continuous self-pollination is done in the plants, there is a chance for what? For inbreeding depression. So in order to prevent this self-pollination, okay, in order to prevent the self-pollination, the plant itself makes some kind of devices, okay, in order to discourage the self pollination and to encourage this and to prevent this inbreeding depression. There are some techniques, okay. The plant itself develops certain techniques, and that techniques will be discussing. In total there are 4 techniques, okay. First technique is the development of or the receptivity of the stigma, okay, of stigma when pollen grains are released. Okay, when pollen grains are released is not synchronized. Release, it is not synchronized. It is not, we can write it as not synchronized. What is the meaning of this? See, you know the structure of a female reproductive structure, right? There is a stigma, then style, and there is an ovary. Okay, so if we are considering the bisexual flowers, the anther will be also present. Okay, the anther will be also present in the same flower. Okay, so if anther and the pistil is present, there is a chance of release of pollen grain towards the surface. Okay, so what is the first method saying? The first method says that the receptivity of the stigma, okay, and the pollen grain release is not synchronized, which means the maturation of the stigma, okay, the maturation of stigma. If you're considering, once again, the stigma is here, then style is there, then ovary is there, okay, and also we have to understand that there is anther and or we can say stamen is present. Okay, so the stigma will mature faster. Okay, stigma will mature faster and anther will release, anther dehisces pollen grains. Okay, anther dehisces pollen grains before the maturation. Okay, before the maturation of stigma, so that the pollen grains which is coming towards this side won't get pollinated. The pollen grains won't get pollinated, thereby it can prevent the self pollination. So we can say the first technique is the not synchronization of the receptivity of the stigma with the pollen release. Okay, so what is the second one? See, this is the position of the pollen grains. Okay, so second one, what will be the second one? What is the second case? Okay, if you are considering the second case, I told you that this is the stigma, then style, and this is the ovary. And if self-pollination has to be happen, the anther should be like this. The position should be like this. Okay, thereby the anther can dehisce the pollen grains, and this pollen grains can be released into this portion where the pollination occurs. This is the condition of self-pollination. Okay, so when outbreeding devices are developed, the second method is change in position. The second method is change in position of pistil and stamen. Okay, pistil and stamen, which means, see, this is the condition where self-pollination can be done. Okay, so if we are considering the change in position, this is stigma, then this is style, and this is ovary. Okay, so here, if anther is present like this, is there any chance to get self-pollination? Is there any chance to get self-pollination? There is no chance of self-pollination. Thereby, it can prevent the self-pollination. That is the second technique. Okay, then we have to study the third technique. Okay, what is the third outbreeding device? The third outbreeding device is self-incompatibility. Okay, third one is self-incompatibility. What is self-incompatibility? Okay, self-incompatibility means it is the inhibition of inhibition of pollen grains. Okay, inhibition of pollen germination. Okay, not grains, it is germination. Okay, it is the inhibition of pollen germination, or it is the formation of formation of pollen tube. Pollen tube in pistil. Okay, in pistil. So this is the third outbreeding device. What is the third outbreeding device? It is self-incompatibility. Self-incompatibility just mean that it is the inhibition of pollen germination or pollen tube growth inside the stigma. Okay, so these are the three different methods. And what is the fourth method? Fourth method is the production of unisexual flowers. What do you mean by production of unisexual flowers? The plant itself undergo a technique known as, you know what is bisexual flowers, right? What is bisexual flowers? Both male and female is present. This male flower and female flower will be present. Okay, both male and female will be present. What is unisexual? Uni means single. Unisexual flowers means only male flower will be present, or male reproductive structure will be present, or female reproductive structure will be present in the flower. Okay, so we can say the fourth method is production of unisexual flowers. Okay, unisexual flowers. So we can say once again, the pollen release stigma receptivity, it is not synchronized in the case of outbreeding device. First, outbreeding technique. Second one, anther and stigma are placed at different positions. Okay, it has different position, change in position. Third one, it is self-incompatibility, and fourth one, it is production of unisexual flowers. Okay, so this is about outbreeding devices. Simple thing, right? Then next we have to study about pollen pistil interaction. Okay, pollen pistil interaction. So see, my dear students, I told you again, this is stigma, style, and next we have ovary. The swollen base is called ovary. The pollen has to come here in the stigmatic surface. Okay, pollen has to come where? It has to come towards the stigmatic surface. Okay, if the pollen grain is viable, if the pollen grain is viable and it has ability to germinate, we will call it as compatible. Okay, if the pollen grain is compatible, it undergoes germination to produce a germ tube. Then later on, it will be coming and fusing it with the egg apparatus present inside the ovary. Okay, so we can say if it is compatible, if a true, like, so for example, see, this is the pistil of hibiscus flower. Okay, this is the pistil of hibiscus flower. So, if a pollen grain of a hibiscus comes and attaches, okay, comes and attaches to the stigmatic surface, what happens? Pollination occurs. Okay, which means pollination occurs. Next one, same hibiscus plant. Same hibiscus plant, and if the pollen grain, okay, if the pollen grain is from jasmine, if the pollen grain is from jasmine, will it pollinate? Will it form any new structure? Will it form a pollen tube? Will it germinate? No, it won't be germinating. It won't be germinating. That means the same species. It should be the compatible. Pollen grain should be compatible towards the stigmatic surface. How it will be compatible? Recently, this researchers have been studied, or the different studies have understood, different studies, from different studies it is clearly understood that different chemicals, okay, different chemicals produced inside the pollen grain, different chemicals produced inside the pollen grain and in the stigmatic surface can form the compatibility, okay, can form the germination, can lead to the pollination and pollen germination. Okay, so next we have to study how the pollen grains is getting towards the stigmatic surface. Okay, they are, how they are pollinating. Okay, so see, this is a pollen, this is a stigmatic surface, then long tube style, and then ovary is present. Okay, inside this there are structures. Okay, inside ovary, you know that there is ovule present, and this ovule is having, inside there is an embryo sac, then we can say an ovule is present with many different stalk. Okay, so this, the ovule is present. So we can say if a pollen grain command attaches to the surface, okay, surface or stigma, pollination occurs. What happens during pollination? In your textbook, it is clearly, when you are studying the structure of pollen grains, we have studied that the pollen grains can be released. Okay, pollen grain can be released in two stages. Okay, one is in two cell stage. Two cell stages. That means they have a generative cell and a vegetative cell. Okay, and also the other condition is it might be three cell. Three cell in cells, not three cell, which is having three nucleus. Okay, so we can say which is having three nucleus. That means it has a generative cell and a vegetative cell. Vegetative cell and a generative cell. The generative cell divides into two, which acts as the male gam. Okay, so this two cell, if you're considering the two cell stage pollen tube or pollen grain, what happens to here? See, if you're considering the structure of pollen grain, this is the structure of pollen grain and it has the germ pore. Okay, it has the germ pore in between. It has the germ pore in between, and we can say there is a generative cell and there is a vegetative cell. Okay, so we can say the germ pore. Through this germ pore, see, we can find the germ pore here, right? This is the germ pore. So we can say during pollination, what happens to the germ pore? The germ pore, the pollen tube. Okay, the pollen tube will be formed through a germ. Oh, okay, there is a pollen tube and pollen tube is formed through a germ. Oh, okay, and inside the pollen tube, what is present? Generative cell. It has two male nucleus. Okay, what it has? It has two male nucleus and this two male gamete. This is male gamete. This two nucleus will be acting as the male gamete. Okay, so we can see the pollen tube which is formed. Okay, the pollen tube. How the pollen tube is formed? It is penetrating towards the tissues of stigma. Okay, the stigmatic. They understood that this pollen grain towards to this particular plant. After that, what happens? The stigmatic surface, to the stigmatic surface, it has to go up, down. Okay, so that penetration of tissues can be done only with the help of pollen tube. Pollen tube grows towards the which region? Ovary. So that is given in this picture. Let's discuss it with the picture, with the help of picture. Okay. So here you can find the pollen grains. Okay, here you can find the pollen grains. And what you can find here, this is the stigma. This is the stigma. And this is the style. Okay, this is the style. And here you can find the ovule. Okay, here you can find the ovule. This is the ovule is present inside the ovary. Ovule is present inside the ovary. There you can find this as the egg apparatus, where you can find three antipodal cells. Okay, then you can find polar nuclei. Polar nuclei. Okay, then you can find an egg apparatus where egg is present, and then you can find the synergids. Okay, so what happens here? See, the pollen tube grows. You can find the pathway of pollen tube. This is the pollen grain, and this pollen grain has germinated and it forms a tube here. Through the stigma style, it enters into the style, then it is moving towards which region? It is moving towards the micropylar region of the egg apparatus. It is moving towards the micropylar region of the ovule, structure of ovule. Okay, so when it reaches to the region called, when it reaches to the micropylar region, so we can say this is an embryo sac where it has a nucellus. Okay, then it has an inner integument, then it has an outer integument, and this is the, we can say that this is the funicle. Okay, so when pollen grain is forming, okay, pollen grain attaches, pollen tube forms. Okay, pollen tube forms inside. Pollen tube is formed through, pollen tube is formed through the micropylar region. It enters into the embryo sac. It enters into the embryo sac. Okay, it enters into the embryo sac. Okay, fine. It enters into the embryo sac through synergids. Okay, so we can draw like this. We can find the synergids. Okay, here the two nucleus will be present. There are two nucleus present, and this nucleus will be entering. Okay, there are two nucleus, or we'll call that it as male gamete. Then it enters into the which part? It enters into the, see, this is the structure, and it enters into the synergid. First, it will be entering into the synergids, and there you can find a structure called filiform apparatus, a finger-like projections inside the synergids. Okay, inside the synergids, what you can find? Synergids. Okay, if you're looking into the synergids, you can find a structure called filiform apparatus. What you can find? You can find filiform apparatus. What is the speciality of this filiform apparatus? This filiform apparatus is the apparatus which guides the pollen tube into egg. Okay, it guides the pollen tube. It guides the pollen tube. Okay, pollen tube into Excel. Okay, into the Excel. It guides the pollen tube into the Excel. So it is very important, dear students. You will get 1 mark question from this filiform apparatus. What is the role of filiform apparatus in pollination? What is the role of filiform apparatus? Filiform apparatus in synergids will guide the pollen tube into the Excel. Okay, don't forget this. So what happens? Now the pollen grain or the male gamete will be fusing with the egg cell, and the next pollen grain will be fusing with the next cell, that is polar nuclei. study that. Before that, we have to study how the planters or breeders are doing artificial hybridization. Okay, now let's see artificial hybridization. How artificial hybridization can be done? Artificial hybridization can be done by the two steps that is called emasculation and bagging. Okay, so if you are considering a flower, a bisexual flower, it may have both the reproductive structures, that is female reproductive structure and the male reproductive structure. Okay, both the structures will be present in a single flower, that is bisexual flowers. Okay, if the female parent, okay, the condition is if the female parent possess, okay, if the female parent possess the bisexual flowers, okay, if the female parent possess the bisexual flowers, bisexual flowers. What is the condition here? Bisexual flower, and bisexual flower means both female and male are present. Male reproductive structure will be present. For example, we can say the stigma, then style, and then the ovary is present. Along with this, the pollen grains. Okay, the pollen grain, or we can say the male reproductive structure called stamen will be present. This is the condition of a bisexual flower. Okay, so what is emasculation? Emasculation is the removal of anther. Okay, what is emasculation? Emasculation is the removal of anther. It is the removal of anther. When anther is removed, what is remaining there? When anther is removed, what is remaining there? Let's remove the anther. Okay. It is completely removed. So we can say when the anther is removed, what is remaining? The remaining thing is only the stigma. Okay, stigma, style, and the ovary. Okay, so we can say what is emasculation? Emasculation is the process of removal of anthers. Okay, removal of anthers, and then we have a stigma, style, ovary, that is pistil here. Okay, so we need to save this pistil from unpollute, or we want to save this from pollution. We can say not the original air pollution, water pollution. We have to remove it from unwanted pollination. Okay, we have to prevent it from unwanted pollination. Okay, pollination. For that purpose, what we have to do, we have to cover this with a butter paper. Okay, we have to cover with a butter paper. So this is a butter paper, butter paper. So the covering of emasculated pistil with the help of butter paper is known as bagging. Okay, so there are two processes in artificial hybridization. What is the first step? It is emasculation. Just understand that it is the removal of anther. Okay, removal of anther using forceps, not with our hands. Okay, it is done with the help of forceps. Okay, after that, the stigma, style, and ovary have to be protected. For that, we are covering it with a butter paper. Okay, what paper it is? Butter paper. Okay, normally butter paper is used. In sometimes we will use the cloth or a small plastic bag. Okay, so that process is known as bagging. Why bagging is done? Bagging is done to prevent the unwanted pollination. Okay, so after when it gets matured, we'll take out this butter paper and the stigmatic surface is free now. Then the breeders will add the pollen grains. Okay, pollen grains, so that pollination can be done. If pollination is done, fertilization will be done. Okay, so it is clear, right? It is clear about this artificial hybridization. Okay, so that's all about artificial hybridization. Then next we have to learn about double fertilization. Next process is fertilization. Okay, so we have learned how pollen grains is entering into the egg apparatus, right? It is moving into the egg apparatus through synergids. Filiform apparatus will guide the pollen tube to enter into the egg apparatus. Okay. So, next what happens? Fertilization occurs. Okay. So, if we have studied for a pollen tube, okay, or for a pollen grain, For a pollen grain, how many male gametes? How many male gametes will be present? Two male gametes will be present. Okay, two male gametes will be acting as male gametes will be present. Okay, so next we have studied about the structure egg embryo sac. Okay, we have studied the embryo sac and also we have learned that they are 7 cell, 8 nucleated structure. Okay, 7 cell, 8 nucleated structure. Right? Yes or no? Yes. So we can say there are two male gametes entering. Okay, there are two male gametes and also let's draw the structure of the female embryo sac here. So this is embryo sac. Okay, if you are considering the embryo sac, see these are the three structures. Okay, these two structures are known as synergids. Okay, then you have an egg cell. Then the central cell, both the cells are known as polar nuclei. Okay, and these three cells together, they are known as antipodal cells, right? Antipodal cells. So what happens during double fertilization? What happens during double fertilization? See, you have studied that with the help of synergids, the pollen tube enters. Okay, the pollen tube enters into the egg cell. Now the egg cell has the nucleus. The pollen also release. The pollen grain enters and pollen grains fuses with the egg cells. Okay, pollen grain also fuses with the egg cells. So we can generalize and write one male gamete. Okay, one male gamete. Okay, we can say that one male gamete. Okay, male gamete and fuses with the egg or the female gamete. Okay, and what is the result of this fusion? What is the result of this fusion? If two gametes are fused, then what will be the result? It will be, obviously, it will be zygote, right? Zygote. Okay, so we can say the formation of zygote. Okay, the formation of zygote by fusion of male gamete with egg. Okay, and that process is known as syngamy. What is the process known as? Syngamy. Okay, the process is known as syngamy. Now, one more male gamete is present, right? The next male gamete, that means we can say another male gamete. Okay, one male gamete. It will be entering into the central cell. Okay, central cell are polar nuclei. Okay, it will be fused with polar nuclei. Okay, it will be fused with polar nuclei. So when it is fusing with polar nuclei, it forms a structure called pen. What is that pen? Primary endosperm. Primary endosperm nucleus. Okay, one male gamete with polar nuclei forms primary endosperm nucleus. We can call this as pen. Not this pen, okay? We'll call it as pen. Okay, so we can say the formation of pen. The formation of pen by the fusion of one male gamete and polar nuclei is known the process is called triple fusion. What is the process called? Triple fusion. Okay, what is the process called? The process is called triple fusion. So, after triple fusion, both the triple fusion and syngamy takes place together. Okay, both this takes place together and we can call syngamy, we can say syngamy along with triple fusion. Okay, syngamy along with triple fusion is known as is known as is known as double fertilization. Okay, is it clear now? Is it clear now? Okay, there are two male gametes. One has to fuse with a female gamete, that is egg, to form a zygote, and that process is known as syngamy. Okay, the remaining one more male gamete is present. That male gamete will be fused with the central polar nuclei to form a structure called nucleus called primary endosperm nuclei. Okay, and this formation of primary endosperm nucleus, the process of formation of primary endosperm nucleus is known as triple fusion, and we can call the syngamy along with the triple fusion is known as double fertilization. I think it is now clear for everyone. Okay, so next question, what happens to the zygote and what happens to the pen? Okay, what happens to the zygote and what happens to the pen? So, my dear students, you have to keep in remember that the zygote, the formed zygote will undergo many division to form the structure called embryo. Okay, what is the structure called? The structure is called embryo. Then you have pen, right? Primary endosperm nucleus, and this primary endosperm also nucleus also undergo division and it forms a structure called endosperm. Okay, it forms the endosperm. Okay, is it clear? Is it clear? Okay, so next we have to study about the post-fertilization events, that is the formation of endosperm, embryo, then seed and fruit. That's it. Okay, so you understood how endosperm is formed and the ploidy of endosperm will be always 3N. You have to please note it down. Okay, please note it down. The ploidy of endosperm will be 3N. So what is the purpose of endosperm? Endosperm is nutritive in nature. It has to give nutrition. Okay, nutrition to the developing embryo. That is the function. Nutrition to the developing embryo. So I can say the polar nuclei, the central polar nuclei, there are two nucleus. Okay, along with this two nucleus, what happens? Our male gamete comes. Okay, male gametes come all together, they fuse together and they fuse together and it forms a single nuclei. Okay, they fuse together and it forms a single nucleus. Okay, now a cell is present. Inside a cell, there is a single nucleus is present. Okay, so what happens to this? How the endosperm is formed? The single nucleus. Sometimes, in the most cases, the single nucleus, at first it will undergo the nuclear division. Free nuclear division will be undergoing. One nuclei, it will be divided into two nuclei. The two nuclei will be divided into three nuclei, then four nuclei, five nuclei, six nuclei, seven nuclei. It undergoes many division. Okay, it undergoes many free nuclear division. That means here what happens? It undergoes free nuclear division. Okay, free nuclear division. Then after free nuclear division, what happens? This is free nuclear division, right? The cell wall will be formed. Okay, what is formed? The cell wall will be formed around this free nucleus. Cell wall will be formed around the free nucleus. Cell wall will be formed. See, the cell wall will be formed and now the endosperm will be in a cellular form. Okay, then what happens? Cell wall forms and cell wall. When the cell wall is formed, it becomes the endosperm. Endosperm becomes cellular endosperm. Okay, what endosperm? It is cellular endosperm. Okay, this is the most of this time this happens in the plants. Okay, endosperm formation is like this. And sometimes this endosperm will be completely utilized by the embryo during its development. In some cases, it is not completely utilized. We can say the example as Coconut, tender coconut. Example is tender coconut. You can find the water, right? You can find the water. Okay. And also you can find the white kernel. Okay. So that white tender coconut inside that can be considered as endosperm. Endosperm. Okay. Endosperm. That is free nuclear endosperm. The water is free nuclear stage. Okay, the water is free nuclear stage. And if you look into the coconut structure, you can find the kernel, right? White kernel. That kernel is actually the cellular, the kernel is actually the cellular endosperm. Endosperm. Okay, it is cellular. White kernel is cellular endosperm, where water is free nuclear endosperm. Free nuclear endosperm. Okay, so what happens here when embryo is developing? The embryo, for the developing embryo, the water will be, or the free nuclear endosperm will be acting as the nourishment after many weeks or many days. Okay, so is it clear about endosperm? You got a clear picture of endosperm, right? Okay, fine. So, next we will be discussing with, okay, next we will be discussing with the structure of embryo. Okay, next we have to discuss with the structure of embryo. Let's see what is embryo or how the embryo is formed. Okay, so we can say after fertilization. Okay, after fertilization, see one male gamete. Okay, it enters into the egg cell and it fuses with the nucleus of egg cell to form the structure called zygote. It forms a structure called zygote. Let's consider this as the zygote. Inside the zygote, there is a nucleus. Okay, so the zygote will undergo division. The zygote will undergo division and it forms the structure called proembryo. What it undergoes and forms? It undergoes and forms into a structure called proembryo. Then it undergoes many division. Proembryo undergo many division to form the structure called globular embryo. Okay, it undergoes a structure called, see, there is a small region like this, then a long suspensor is present, suspensor cell is present. Okay, then the suspensor cell, along with the suspensor cell, we can find a globular part. And this globular part, okay, this is globular part where it contains many different cells. Okay, this is the globular embryo. So we can say this is the globular embryo. Okay, this globular embryo it undergoes division and forms a heart-shaped embryo. What shaped? A heart-shaped embryo. It forms a heart-shaped embryo. See again, it undergoes many division. Undergoes many division. It has many cells. It has many cells. Okay, and the shape of the embryo is heart-shaped. Since the shape is heart-shaped, we can call it as heart-shaped embryo. Okay, so from globular embryo, it forms heart-shaped embryo. So from heart-shaped embryo, it forms a complete embryo. It forms a complete embryo. Okay, it forms a complete embryo. That means a mature embryo. So these are the major phases of the formation of embryo. So you have to study this for 2 marks or 1 mark. there is a chance of asking this question. Okay, so what is the condition? Pro-embryo. First it was zygote. It undergoes division and forms pro-embryo. Pro-embryo undergoes division and forms globular embryo. Globular embryo undergoes division and forms heart-shaped embryo. Heart-shaped embryo, it gets matured and forms mature embryo. Okay, it gets matured and forms mature embryo. You have to study this, study this, study this. Okay, see, this is the zygote. Then zygote undergoes division to form globular structure, globular embryo. See the swollen base. Base you can see like a globe structure, right? That's why we will call it as a globular embryo. Then here the shape of this embryo is transformed into a heart-shaped structure. So we'll call it as a heart-shaped embryo. Then a mature embryo is formed. Okay, so we can say the general formation of both the embryos in monocote and dicote are same, but the structure is little bit different. Let's see the structure of dicote embryo. Okay, let's see the structure of dicote embryo. So this is the structure of dicote embryo, where we can say there are different parts known as, in embryo we have embryonal axis. We have embryonal axis. So the part above the embryonal axis. So if you are considering this as the embryo, see this is in a twisted portion. Like let's see, let's keep it in an above condition like this. Okay, like this. This is two cotyledons and this is the embryonal axis. Okay, let's see. This is the embryonal axis. If you are saying, if we consider, okay, if we consider the portion above the embryonal axis, we will call it as epicotyle region. What we will call it is epicotyle region. Okay, then we will call the below the axis, below the embryonal axis, we will call it as hypocotyle region. Okay, below the embryonal axis, we will call it as hypocotyle region. Okay, so embryonal axis, if the epicotyle is ending with a structure and that structure is known as plumule. Okay, and that structure is known as plumule. See, here you can find the structure. This is plumule. Okay, then these are the two cotyledons. These are the two cotyledons. Okay, the embryonal axis above the region above the embryonal axis, we'll call it as epicotyle region. Okay, the region below, we'll call it as hypocotyle region. Okay, the hypocotyle ends with radicle. The hypocotyle ends with radicle, or we'll call it as root tip. We'll call it as root tip. Okay, then there is a small covering for this root tip and we'll call it as root cap. We'll call it as root cap. So this is the structure of dicote embryo. Okay, you have to study how to draw the dicote embryo. Okay, this I just draw this to make you understand that there is an embryonal axis. Actually, this is the image. You have to study this image. Okay, not this one. Okay, so fine. Next, we will be discussing with monocote embryo. Okay, so monocote embryo is also having the same structure. Not we can say only one cotyledon is present. How many cotyledon is present? One cotyledon. In the case of dicote embryo, you understood that there are two cotyledons. Two number of cotyledons are present. But in the case of monocote embryo, only one cotyledon is present. Okay, so the structure will be like looking like this. Okay. Okay, the structure looks like this. So we can say this is the cotyle region. Okay, this is the cotyledon. This is the cotyledon. So if you are considering the grass species, the cotyledon is known as scutellum. Okay, the cotyledon is known as Scute alla. Then you study that is there is a shoot tip. Okay, there is a shoot tip present, and this shoot tip is enclosed in a hollow foliar structure, and that hollow foliar structure is known as coleoptile. Okay, that foliar hollow structure, that hollow foliar structure is known as coleoptile. Then you can find this, you can find the shoot tip, shoot tip. Okay, shoot tip here. Then you have, root tip. Okay, root cap. Then you have a root tip. Okay, root tip. Then you have root cap, and those root tip and root cap is enclosed in a particular structure, and that structure is known as coleorhiza. Okay, coleorhiza. And this is the root cap. Root cap is present. Then root, or we can call it as radical is present. Okay, we can say like the same one. There is an epicotyle region above the axis. Above the axis, we will call it as epicotyle region. Okay, epicotyle region. Below the axis, we will call it as hypocotyle region. What we will call? We will call it as hypocotyle region. Okay, hypocotyle region. Okay, so there is a small outgrowth like structure here, and actually it was a cotyledon, but it gets into, it transformed, or we can say that it is a rudimentary part of that cotyledon, existing cotyledon, that we will call it as epiblast. Okay, that small structure we will call it as epiblast. This is the structure of a monocot embryo. Okay, this is the structure of a monocot embryo, which we have just discussed. Okay, so next we have to study about seed. Okay, here I have mentioned what is a seed. Actually, seed is, we'll call seed as fertilized ovule. Okay, what we'll call? We'll call it as fertilized ovule. And what are the different portions of a seed? Seed has seed coat, and it has cotyledon, and also it has embryonal axis. Embryonal axis. Okay, so seed has seed coat, cotyledon, and embryonal axis. Okay, so we'll classify the seeds into two different groups. Okay, we'll classify the seeds into two different groups, that is albuminous seeds and non-albuminous seeds. Albuminous seeds are those seeds which has endosperm in it. Okay, albuminous seeds are those seeds which has endosperm in it. Okay, next example I have mentioned before, that it is coconut. Coconut is there, then castor is there. Okay, wheat is there, then what else? These are the different examples for albuminous seeds. Okay, next we have to study about non-albuminous seed. What are non-albuminous seeds? Non-albuminous seeds are those seeds which is not having no endosperm, which is not having the remnants of endosperms. Okay, which is not having the endosperms. For example, we can say pea. Pea is an example. Beans. Beans is an example. Then groundnut. Okay, groundnut. These are the examples for uh, non-albuminous seed. Okay, then last one, it is perisperm. Okay, what is perisperm? See, we have the structure known as, this is egg apparatus. Egg apparatus, then covering egg apparatus, there is nucellus, then outer integument and inner integument, then a funicle is present. Okay, so this is the nucellus, right? This is the nucellus. If nucellus is remaining, okay, if nucellus, okay, if nucellus is remaining in the seed, okay, then that kind of seed we will call it as perisperm. Okay, we can say in perisperm, remnants of nucellus is present. nucellus is present. Example, example, black pepper. Okay, what is the example? Black pepper is the example. Okay, got it? Understood? Okay, so next we have to study what happens to the outer covering. Okay, see I told you that it is fertilized ovule, we will call it as, what we will call fertilized ovule as the seed. Okay, so the outer covering is present, the outer covering, what is the outer covering? Outer integument. Outer integument will be acting as the outer seed coat. Okay, outer integument, it will be known as outer seed coat. Okay, then you have inner, this outer seed coat will be tough structure. They are tough in nature, right? They are tough in nature. Okay, then you can understand that the inner integument. Inner integument is the inner seed coat. Okay, it is the inner seed coat. Inside this seed coat, both the seed coats, what is present? This embryo. Egg apparatus, it will be converted into embryo. Structure called embryo. Nucellus will be present. That nucellus was to give nourishment to the developing egg apparatus. But when it fuses, it forms embryo. And for the nourishment of embryo, what is present? Endosperm is present. Okay, you have to understand the difference, structural differences. Okay, so this is how the ovule becomes the seed. Okay, now my question, where does this ovule is present? This ovule is present inside the ovary. The swollen base known as ovary, right? So what happens to the ovary after fertilization? Ovary will be turned into a fruit. Okay, so what is a fruit? Actually, ovary is transformed into fruit. Okay, so we can classify the fruit into three groups. What are the three groups? False fruit, true fruit, and parthenocarpic fruit. Okay, let's see first false fruit. What is false fruit? The fruit which is developing apart from, okay, fruit which develops apart from ovary. Apart from ovary, we'll call that fruits as, apart from ovary, we'll call that fruit as false fruit. Okay, so what is the example for false fruit here? We can say apple is an example. Okay, apple is an example. Fine. Then true fruit. How apple is formed? It is formed from thalamus. Okay, it is formed from thalamus. Okay, so that is about false fruit. That means simply it means if the fruit is formed rather than from the ovary. Okay, like thalamus or from the other parts of the flower, we can call it as a false fruit. Okay, so next one, it is true fruit. Okay, what is true fruit? If the fruit is formed from ovary, if the fruit is formed from ovary, we'll call it as a true fruit. Okay, what is the example here? Mango is an example. Mango is an example. Mango is so yummy, right? So we can say that is a true fruit. Okay, last one, it is parthenocarpic fruit. What is parthenocarpic fruit? It is the formation of fruit without fertilization. Fruit without fertilization. Okay, fruit without fertilization is known as parthenocarpic fruit. And you have to remember that always these fruits are seedless fruit. Always these fruits are seedless fruit. Okay, also remember the example. Example is banana. Do you find any seeds in banana? No, right? So we can say banana is a parthenocarpic fruit. Okay, clear, understood, done. Okay, so these are the three different types of fruit, that is false fruit, true fruit and parthenocarpic fruit. Next we will be discussing with, these are the false fruit. Okay, so apomixis and polyembryony, this is the last portion of this chapter. What is apomixis and what is polyembryony? Okay, let's discuss first, we will discuss with apomixis. Okay, apomixis. It simply mimics the reproduction. Okay, mimics the sexual reproduction. Okay, it mimics the sexual reproduction. That means simply we can say it is the mode of reproduction where there is no fertilization. Okay, we can say formation of seed without fertilization. Okay, seed without fertilization is known as apomixis. Formation of seed without fertilization. Okay, said it is known as apomixis. Okay, next we have to study as polyembryony. Okay, last topic it is polyembryony. So what is polyembryony? It is from the word itself you have to under polyembryony. Okay, polyembryo or polyembryony. What is the meaning of polyembryony? Okay, what is the meaning of poly means many. Okay, embryony means it is a formation of embryo. Okay, so you know the structure again. I am drawing the structure. This is egg apparatus. Around the egg apparatus, there is nucellus. Okay, inside the egg apparatus, there is synergids, then antipodals, and the polar nuclei. Okay, so if the nucellus cells, okay, if the new cells of the nucellus, if the cells of the nucellus is forming embryos, inside the egg apparatus, rather than from the normal fertilization process. Okay, rather than from the normal fertilization process, here many embryos will be formed. Okay, many embryos will be formed. And if many embryos are formed within a single ovule, many embryos are formed within an ovule, then we will call it as polyembryony. Okay, we'll call it as polyembryony. Where you can find the polyembryony? Can you give an example for polyembryony? It's a common example. Okay, what are the example? Citrus is an example for polyembryony. Okay, then we can say citrus, lemon. Lemon belongs to the citrus family, right? Lemon is an example. Orange is an example. Orange is an example for polyembryony. Okay, so these are the different topics that we have covered in this chapter. Okay, so I think you are very much clear about the topic, clear about the contents, right? So in this chapter, from the beginning, if you are looking, we have discussed about the structure of both male reproductive structure and female reproductive structure. Then we see how microsporogenesis occurs, how megasporogenesis occurs. Then coming to the next part, that is fertilization, fusion. Okay, pollination we have studied. Then fertilization, double fertilization we have studied, and what are the different post-fertilization events. These were the contents that we have discussed here. So next class we will be discussing with another new interesting topic. Until then, stay tuned. Thank you.